Hi bio students, welcome to lab doing DNA extraction from strawberries. So you're going to need some fresh strawberries. Take the stems off, you need 3 of them. You're also going to need a mixing bowl, measuring cup, some measuring spoons, strainer, table salt, liquid dish soap, Ziploc baggie, a glass or a beaker, and some alcohol. This is ethanol, 70%. That was in the freezer. So 70% alcohol is like rubbing alcohol, and keep it in the freezer. Okay? So first step is to take a half a cup of water, put it in your bowl, and then to that I'm going to add a half a teaspoon of salt and 1 tablespoon of liquid dish soap. So you want to give that a good stir. Stir it up. Okay. After you've stirred it up really well, you're going to take your Ziploc baggie, add in your 3 strawberries that you took the stems off already, and then 3 tablespoons of the liquid that you just mixed up, the dish soap, salt, water mixture. So 1, 2, 3. So you seal it up, get all the air out of there. And now we need to mash them up really good so it's all mush. So just start squeezing the berries. You basically want to break open those cell membranes, right? Because all the DNA is inside of the cells of the strawberry. Now, I bet you remember that DNA is in the nucleus. So we have to break open the cell wall, the cell membranes, and the nuclear membrane to release the DNA. Now that's why we added the dish soap. The dish soap is a very important ingredient because it's a lipid, and it's gonna it breaks. You know how when you add dish soap to like when you're cleaning a frying pan, it gets all the grease off the pan? Well, when you're adding it to the strawberries, the dish soap is helping to break apart the lipid membranes of the cell membrane and the nuclear membrane, which they're made of lipids. So the soap breaks apart the bilipid membranes. Now, I said the soap was a lipid. I take that back. It's breaking open the lipid membranes. Okay, so I'm mixing it all up, and I'm squishing it. I'm gonna break these cells open. You can see it's looking pretty juicy and red. And our next step is we want to filter. We want to filter to get all that pulp. We don't want all that strawberry chunks to go into this. We want to get just the liquid and the DNA to go through my strainers. Put your strainer on top of your glass, and you want to make sure your strawberries are all really broken up really well. Then you pour it through the strainer. It's kind of foamy. Now it might take a while for it to filter through the strainer, but you can use your spoon to kind of push it through the strainer. Some chunks there. I'm gonna use my spoon to kind of like get some of more of that liquid. You want to see the DNA of the strawberry. And you can really use this technique on a lot of different fruits or vegetables. Bananas work well. I've done this with split peas before. I used a blender to break apart the cell walls and cell membrane because I couldn't break it with my fingers, of course. And we've done it with other veggies as well. So now I got all this pulp, and that's going to get thrown out. But now what you want to do with your ice cold ethanol, you're going to pour this, and I have written down it takes about 1/3 of a cup or so. Basically, this alcohol is used to layer on top of the strawberry liquid, because when it makes a layer on top, then the DNA can float up into that clear layer because the alcohol is clear. So I'm going to tilt it and just run it down the side So I don't want them to mix together. I want them to slowly pour the alcohol on top to make a clear layer on top of the strawberries. Nice and slow. Okay, now when you look here, you can see that there's some stuff that looks like kind of like almost like mucus. It's like a whitish color, and it's above the strawberry in the clear alcohol layer. The stuff that's floating up to the top, that's your DNA. It's coming out of the solution at the bottom. And then once it does that, you'll be able to see the DNA nice and clear. I'm not sure how well you can see this. I'm trying to hold it close, but there's a lot of DNA that just floated up into this area right there. So if you have something like tweezers, it'd be kind of cool where you can kind of take a stick. See that? That is DNA out of many, many thousands of strawberry cells. Looks like snot, but it's not. Okay, so isn't that cool? Everything in this world has got DNA, and it's universal way of storing blueprints of life. And it's just neat how you can do a simple experiment and then see what DNA actually looks like. Now, this DNA may still be wrapped around some histones, little proteins, but that's not from one cell, that's from thousands and thousands of cells. Okay? Well, I hope you guys had fun with this experiment, and I'll talk to you later.